All right, so we are now looking at alternative forms of inheritance. This is, luckily for Mendel, he didn't run into any of these because that would have skewed his data and he probably would not have been able to figure out what was going on. Maybe, but probably not. But now we know that not everything is simple, straight, what we call complete dominance or simple dominance or normal Mendelian inheritance. And so these are some of the others. We're going to talk about one more, I think, next week, which is sex-linked. But these are some of the others that we've talked about this week. So the first one is incomplete dominance. So think about what it means to be complete, like 100%. Incomplete means that it's not 100%. And what this is going to look like is the red says, well, I don't really want to be dominant, and the white says, well, I don't really want to be dominant either. And they say, well, okay, well, why don't we just split the difference? Why don't we compromise? And so this is looking at flowers. There's other examples too. This is just the most famous, common, whatever you want to say, example. But in these flowers, if they have two CR genes, this would be a red parent. CWCW would be phenotypically white. It would be, when you looked at it, it would be white. So genotype, phenotype, genotype, phenotype. Okay, and then if it's a mix, if it's a heterozygote, has one of each, then it's going to be pink because that's what happens when you mix red and white. And so before Mendel, they had this blending theory that basically said that mom's dads and Jean's dad's genes blended together and the kids came out kind of in between. And that's exactly what incomplete dominance is. So while they were wrong, they were right in this instance, but that's not, they didn't know that. Like they thought that's how it happened 100% of the time. And Mendel came along and said, no, there's these pieces of something that are getting inherited. Now we know those are pieces of DNA called genes, but he didn't know that. But anyway, so the blending theory was wrong, but now we know that sometimes it kind of does happen that way, and that's what incomplete dominance is that the heterozygote is somewhere in between the either parent. If it was yellow and blue, then the heterozygote would be green, because that's what happens when you mix yellow and blue. And so here's a Punnett square. So the other thing is I can define it, and the second is I can model it. And for this, modeling is doing a Punnett square. And so what you should be able to do is you should be able to say, if they say one parent's red and one parent's pink, then you should know, okay, red, the only option for red is two red genes, and the only option for pink is a red and a white. And so you should be able to put the parents on the outside, fill in the children, okay, so whatever parent joins with whatever parent, and then you should be able to decipher that back into the phenotype. So you write your genotype ratios, and then you would know that, hey, this is red, this is pink. If there were any CWCWs, that would be white. And so we have two reds, two pinks, and no whites, or 50%, 50%, 0%. Okay, so that's how you would model incomplete dominance. You may want to practice some others. Maybe a white times a red, or a white times a pink, or two pinks. Two pinks would be a good one to try, just to see if you can do it. And you can always send me those answers if you want me to look over it and see if you got it right. You did do a worksheet too, so that's another way you can check and make sure you're doing it right. All right, the next one is codominance. Codominance, these are kind of like too nice, and they're like, well, you be dominant. No, you be dominant. Okay, we'll be dominant. We'll kind of half it. Each of us will be kind of half dominant. These guys are bullies. I want to be dominant. No, I want to be dominant. And so their compromise is that they both get to show up in their full glory. So again, my favorite example is the chicken. It's just so cute. CB is a black feather gene. CW is a white feather gene. And so what we have here is we have two heterozygote chickens that are mating. And these heterozygote chickens, you can go back and look at the PowerPoint. They're really cool looking. They're speckled. So they actually have black feathers and white feathers. They don't have gray feathers. That would be incomplete dominance. But they have fully black feathers and fully white feathers side by side. And so when they mate, they're going to get 1/4 chance of a black chicken, 50% or 2/4 chance of a speckled chicken, and 1/4 chance, CWCW, of a white chicken. So 25% black, 25% white, 50% speckled. Okay, and so that's codominance. They're both going to show up in their full glory. There's no mixing, there's no blending. All right, the next one, I can define multiple alleles. Multiple alleles is simply where you have more than two options. Normally we have tall and short, brown and blue. Whatever. You can roll your tongue or you can't. You've got a widow's peak or you don't. But sometimes there's more than two. And so this example with the rabbit's fur is one example. Blood typing is another. There are 3 alleles, A, B, and O. The thing about multiple alleles is each individual still only has 2 alleles. You can't have more than 2. You get one from mom, one from dad. There's no way to have more than that. And so with blood typing, even though there are 3 options, A, B, and O, you're only going to have 2 of those. You could have 2 A's, 2 B's, you could have an A and an O, B and an O, an A and a B, or 2 O's. Same thing with these rabbits. They have 2 alleles. The one that I did earlier, I picked 2 heterozygotes, a full-colored. And again, they will always tell you the order of the hierarchy of dominance. So this is the most dominant. It's dominant to all the others. Chinchilla is the second most dominant, meaning it's dominant to these 2 but not full. Himalayan is the next most dominant. It's dominant to albino but not these 2. And then albino is the recessive. So it's recessive to all the others. And so what we did is we mixed a full and chinchilla heterozygote, which would be full colored because full is dominant, with a Himalayan albino heterozygote, which would be Himalayan because Himalayan is dominant to albino. And out popped these kiddos. So we did our genotype ratio, 25% each, and then we converted that back up to the phenotype. So we know that because this has a big C, it's full. This has a chinchilla and a Himalayan. Well, chinchilla is dominant to Himalayan, so that would be a chinchilla. This has a big C, so it's full, so that's our second full. And then we had a chinchilla and an albino, but chinchilla is dominant to albino, so we know that that would be another chinchilla. So even though our parents were full and Himalayan, our kids are going to be full or chinchilla, 50-50 chance. Okay, so really multiple alleles is done very similarly to just a regular monohybrid because you do have a dominant and a recessive. It's just that you have more than 2 options to choose from when you're coming up with your gametes for your parents. All right, and the last one is polygenic inheritance. And so the one that we looked at was skin color. If you only have 2 genes, you could do a dihybrid cross, 16 squares. If you have 2 genes, you can do the rules of probability. If you have more than 2 genes, you're going to want to do the rules of probability. I'm sure there are people out there that can do a trihybrid. I am not one of them. And so I can define polygenic inheritance. Poly means many, genic is gene. So this is when you have more than 1 gene contributing to a phenotype. For this, you're generally going to see a range of traits. So when you look at something like skin color, there's, gosh, I don't know, 100 different kinds of skin color. It's not just 2 or 3. And so that's what tells you it's more than one gene. Eye color, hair color, height. You know, we don't just have 5 feet and 6 feet. We've got all these different heights. All of those things are polygenic in nature, and we know that because there's just a range of traits. There's a bunch of different options for skin color, hair color, height. So anyway, what you'd do is you would get the parents and you would run a Punnett square with each letter separate. So we have big A times big A, little b times big B, little b, and big C times big C, little c. And you come up with the results. And generally with a polygenic, they're going to ask you for a specific kid. What is the chance of having this kid? And so you would pull those numbers, those fractions, or you could do percentages, and multiply them because this is this and this and this. So and means multiply. And so in this case, it was a 1/8 chance. And so that's how you would model a polygenic inheritance. The last one with this, and I kind of went out of order, is doing a blood type. And I will do one more of those because sometimes those are a little tricky. So the first thing is you need to know this chart. You don't necessarily need to memorize it. On the AP exam, they would give it to you. You may use it enough that you kind of have it memorized just from doing it. We have done homozygous A and heterozygous A. Same thing with B. What we haven't done is an AB times an O, so that's what we're going to do this time. So we have IA times, IB times O. And it's going to give us that. Okay. Genotype ratio, we're just going to write what's there. There are 2 of each. Okay, you could also go 50-50 if you wanted to. And then, like I said, with phenotype, I like for you to do them all. This would be A. This would be B. No ABs, no O's. And so I can set up and solve a blood typing Punnett square. Again, make sure your parents are correct so that your kids are correct, and make sure you can do your genotype and phenotype frequencies with both numbers or percentages. So again, 50-50, 50-50, 0-0. And that's it. I hope that helps, but if you've got questions, let me know.